Hi, my name is Tim Liu. I'm a professor at MIT. I'm here to tell you today about synthetic biology, an emerging engineering discipline. One of the challenges in defining the term synthetic biology is that it's a term that's used to mean different things to different people. And I've listed several of the common definitions that people use for this particular term here on the slide. Regardless of how you exactly define synthetic biology, I think this is a community that's unified by the desire to engineer biological systems for new functions, whether we're doing this from the ground up, from minimal systems, or from the top down, taking an existing system, trying to retrofit it to do new things. This is a robust and vibrant community that is growing. I come from the community at MIT. We have the Synthetic Biology Center there, where we're actively engaged in various aspects of engineering biological systems. Globally, there's a growing community of labs interested in the field of synthetic biology, and this is well evidenced by the iGEM competition, which has grown from a very small community to a very large global community that comes together every year to compete in terms of genetically engineered machines. From an engineer's perspective, one of the exciting things about the field of synthetic biology is that we're moving from an era where we were simply cutting and pasting DNA from one place to another to a point where we're able to specifically program the A's, T's, G's, and C's that go into a biological software. And in some sense, this is really a technologically driven improvement. So here, this is a graph showing a comparison of Moore's law, shown in blue, which is named after one of the founders of Intel, Gordon Moore. And he basically predicted that the semiconductor industry would be able to double the number of transistors on a given chip every 1 or 2 years. And this exponential scaling in computing power has led to tremendous advancements in our ability to program silicon-based systems. Now, if you look at the other two curves on this graph, you can see that our capabilities for DNA sequencing and DNA synthesis are also increasing at an exponential rate. And therefore, our raw capability to program and read DNA is increasing every year. That's really giving us the raw capability to modify biological systems to do new functions. However, there are multiple challenges that are associated with engineering biological systems that are very different from conventional ways of engineering electrical circuits. So for example, in electrical circuits, you can take components and explicitly wire them together to achieve new functions. Whereas in biological systems, parts that we develop are bouncing together in the cell and really are wired based on chemical specificity rather than physical wiring. And this poses a challenge when you're trying to scale and build complex biological circuits. As a result of this implication, in electrical circuits, you can actually duplicate the same identical device, the same transistor, over and over on a chip, and wire these identical devices together to achieve some more complex function. Whereas because biology requires specific chemical wiring between components, you need to develop unique devices to implement every additional level of complexity that you're trying to achieve. Electrical circuits have had many years to try to develop formal languages and designs that allow you to compose more complex functions, whereas the field of designing biological circuits is in its relative infancy, where there are still new concepts and new circuit designs being developed every day, and therefore no kind of dominant formal language being present to describe how to program biological systems. The dream of the field is really to start from the bottom up, to be able to program the DNA code, and to start developing devices and circuits and modules that have additional layers of complexity, with the ultimate dream of to build very complex systems and networks, something like a whole organism that carries out a specific function. We're still quite away from the ultimate dream of kind of engineering a very complex system, but we have made a lot of progress in terms of building devices and circuits and starting to layer these together into higher order modules, which I'll describe in my talk today. The idea I'd like to communicate to you today is that biological circuits are at the core of synthetic biology because they enable complex sense and respond behaviors from cells. So cells can be thought of as little mini computers in that they sense all sorts of information. They process that information so they can perform computations, but they can also store the output of these computations in memory. And then the cells can respond by carrying out new functions, whether it's deciding to live or die, or divide or not, or produce a certain molecule. So at the core of this, we need the ability to program biological systems to compute information and to store this information in memory, because this will enable us to build very generalizable frameworks for sense and respond behaviors. The other thing I'd like to talk about towards the end of the talk is really how far we are in this field from being able to rationally program biological systems that carry out specific functions. So we'd like to really adopt the engineer's framework where we can go from concept to design, model, simulation, where we kind of develop our designs in silico. Then we experimentally construct, probe, and measure these systems and iteratively improve our systems. And I'll talk about some of the design challenges that are still remaining in the field that make it challenging to implement this entire pipeline. The other interesting thing about biology is that unlike silicon-based systems or other engineering disciplines, we have the added capability of evolving biological systems. And that could present actually interesting ways of trying to design systems that carry out new functions, but also poses a challenge when you're trying to design a biological system that carries out a function. And we want the cells to continue to develop that function or display that function without evolving away from it due to the other evolutionary pressures. And so this is a unique challenge, but opportunity for biology, this idea that you can incorporate evolution into the design process. So as I mentioned, there are many flavors of synthetic biology, and even in the field of circuit design, there are many reasons why people are trying to design biological systems. For example, people are interested in solely the idea of computation. Can you design circuits that can allow you to compute functions inside of living cells? Other folks are interested in this idea of therapeutics. So if you could design cells that carry out a specific function, could you use that to program new cell and gene therapies that are responsive to their environment? Other folks are interested in producing new molecules. So if you could design circuits that can sense metabolites or sense intracellular state, could you optimize the expression of new heterologous materials that cells are making, such as chemicals or other physical materials? And finally, there are folks who are interested in this idea of using genetic engineering or synthetic biology to build circuits to understand biology at a deeper level, with the idea that if I cannot build a system, then I cannot fully understand it. And therefore, people are interested in building genetic circuits as a way of understanding basic principles of how biology functions. So before I get into the deep circuit engineering aspects, I wanted to introduce the concept of sensors in synthetic biology. And so many of the circuits I'm going to describe to you later on take in either chemical or light-triggered inputs. There are additional inputs that cells can take in, but I wanted to focus on chemicals and light because this allows us kind of a very flexible set of inputs that you can put into cells. And so there have been a lot of work to develop a variety of different sensors, either at the transcriptional level or the translational level or at the protein level, that can arbitrarily bind to different chemical inputs and trigger gene expression downstream. And what this allows you to do is to then take these inputs. For example, here we have salicylate and arabinose as two different inputs to a circuit that actually calculates an AND function. And that allows us to then build cells that only respond in the presence of both of these inputs because of this logical AND circuit that comes downstream. And so these inputs you can think about as ways of inducing changes inside of the cell, the kind of like little knobs that you can use to turn on and turn off gene expression inside the cell. And then for real-world applications, you can think about these inputs or sensors that can detect real-world analytes or chemicals, such as biomarkers of disease or small molecule chemicals in the environment that you're trying to detect. These can all be hooked into the genetic circuits that I'm going to talk about later on. Another set of sensors that have been used to quite good success has been light. So here's an example from the lab of Chris Voigt that have developed optogenetic sensors in bacteria that can trigger the production of a pigment from these bacteria. And what this allows you to do is to actually program cells to be responsive to light. And here's an example where they've made a mask of Albert Einstein and shown light on a plate of bacteria and basically developed a bacterial photograph by triggering gene expression based on light input. And so light is another very interesting knob by which you can use to turn on and turn off gene expression, with the advantage that you can program multiple orthogonal inputs to a cell by using different wavelengths of light, and you can control dynamically and spatial temporally the gene expression by basically turning on, turning off light at different locations or with different timing. Now, as I mentioned, there are other sensors that you can plug into genetic circuits. And so what I want to tell you now is about different genetic circuits that you can actually start layering on top of these different types of inputs. So in our lab, we're quite interested in two forms of computation, digital and analog computation. So digital computing is kind of the classic computing that you think about when you think about silicon-based computing, like the computers that you use. And this is where you take a signal, whether it's a voltage or chemical concentration, and you split it into zeros and ones, and you only compute based on zeros and ones. There's also the idea of analog computing, where you don't discretize the signal to zeros and ones, and you basically compute using continuous values. And what we've done in our group is to split up both digital and analog computing into two different categories. One where you have memory, where you basically store the output of a computation, and then you also have computing, where you calculate specific functions. And so in the digital case, I'm going to talk about how you can build digital logic gates that calculate Boolean logic functions, and simultaneously store the output of those calculations in DNA-encoded memory. And the ability to integrate both logic and memory together really gives you more powerful computing powers. I'll also touch briefly upon analog computing, so how you can engineer cells to compute mathematical functions based on continuous chemical concentrations. And finally, how you can build memory circuits that actually record analog memory. What I mean by analog memory is when you want to encode more than just absence or presence information into DNA-encoded memory, but encode something like how long or how much of a signal you've seen. So just to summarize again, digital computing is this idea where you take a signal. In the case of biology, this might be a chemical concentration, and you split it into zeros and ones. So you set thresholds, and you say everything below a certain threshold is a 0, everything above a certain threshold is a 1, and we basically compute solely based on zeros and ones. And the reason why we might want to do this is that digital computing allows you to take zeros and ones and build together more complex logic functions like Boolean logic gates. Here we have an example of an AND gate, where you can very easily reason what the input-output relationship might be. So for example, in an AND gate, the output is true, or a 1, only when both inputs are true. And the reason why we might want to do that is that there's a whole framework developed where you can take these individual simple logic gates and piece them together, as shown here, into more complex systems. Here, this is an example of an adder that is composed of multiple logic gates hooked together in a defined way. We would argue that in biology, some systems perform in a digital-like fashion. So if you think about a cell making a decision whether to live or die, that's essentially a digital decision because basically cells can be in one of two states, alive or dead. Or the idea of cell differentiation, where a cell starts in a single state but then differentiates into multiple downstream states, is another example where this digital idea, where you have discrete states, may make sense. So there have been efforts in the past to build digital logic gates and memory circuits. Here's one example where folks built a genetic toggle switch based on two cross-coupled transcription factors, each of which represses the other. And what this allows you to do is create a genetic toggle switch where either one transcription factor is high and the other one is low, or the other transcription factor is high and the other one is low. And you can switch between these two states by adding an input. For example, in this case, one input is heat, the other input is a small molecule, IPTG. And what that does is that it inactivates one of the two repressors and allows you to flip the switch. So this is one way of encoding digital memory inside of a living cell. This idea that you can use two cross-coupled transcription factors, very similar to how the bacteriophages encode a switch, or in electronics, how people encode a set and reset latch. Another way of encoding digital information is to use a class of proteins known as recombinases. And what these recombinases do is they can actually flip a piece of DNA upside down. So if you imagine DNA as kind of a memory device where you can specifically address locations on the DNA and flip them from one orientation to the other, this allows you to store 0 or 1 information in the orientation of DNA. So here's an example where we have a sequence of DNA here that carries a specific orientation. We can define this as the off state. And then if we can express a recombinase that flips the DNA upside down, the DNA has now adopted another sequence because it's inverted. And we can define these two distinct sequences as distinct from each other, one being 0 and one being a 1. So in one state we have off, and the other state we have on. And the nice thing about this approach is that because we have a single piece of DNA that can be in any one of two orientations, you could naturally have digital behavior. So the DNA can only be in one of two orientations. This platform is potentially very extensible. For example, there are many known recombinases that are available that you can use to encode multiple bits of information in DNA. And as I'll show you, this system is modular. So you can put together multiple recombinase-based switches together to build higher order circuits. So here's just an example. Imagine you have a stretch of DNA where you have 3 different sequences, red, blue, and green. And each one of these is targeted by a different recombinase, and they have a specific sequence. So if we could express a recombinase that only targeted the blue sequence and would flip the blue sequence upside down, now we've changed the blue sequence from a 0 to a 1 because we've changed the sequence that's intervening between the blue recombinase recognition sites as shown here in brackets. Now if we express a green recombinase that targets the green brackets, the green recombinase recognition sites, and flips that DNA over, this would allow you to then encode another bit of memory into the DNA by flipping the 0 to a 1 in the DNA that's located between the green sites. So this is a way by which we can encode multiple bits of memory into DNA. And because DNA is passed down stably from mother to daughter cells, this allows you to pass on long-term memory down a chain of living cells and encode memory. So now how can we use this to encode higher-order functions? One of the things we like to do is to be able to encode integrated digital logic with memory. So we want the ability to store information in DNA, but we also want the ability to compute digital logic functions like Boolean logic gates. And so for example, we want the ability to build a Boolean logic gate that's shown up here where you have an AND gate that has two inputs, A and B, and has an output that's only 1 or true when both inputs are 1 or true. So we want people to be able to dig into a set of predefined parts to assemble these circuits in a very easy way. So in this example, we've actually used two different recombinases, the blue recombinases, which target these blue brackets as shown here, and we have red recombinases that target these red triangles. And then sandwiched between these recognition sites, we have actually a variety of gene regulatory elements, including transcriptional terminators as shown here by the T. We also have promoters as shown here by the arrows, and we have genes like GFP that represent the output. The idea then is that we can assemble these regulatory parts into a linear stretch of DNA as shown here that implements the AND gate. And this circuit will obey this following logic function. The output, which is GFP here, should only be true or on or expressed if at least one promoter, so one of these promoters is in the upstream upright orientation, and if none of these terminators are active, so for example, when the terminators look like a T, they're active, so they block transcription, but when they're upside down, they're no longer active. And when the output gene is in the right orientation, so it must be upright, so it can be expressed from the upstream promoter. And as I'll show you, this allows you to implement every single Boolean logic gate that you might want to build. It also allows you to program things like an AND gate, and I'll walk you through an example there. So here's an example of the AND gate I showed you before. It has a GFP, which is the output gene. It has a promoter upstream, which drives transcription, but it has two intervening terminators that block the activity of that promoter. And each of these two terminators are sandwiched in between a recombinase recognition site. For example, this terminator is sandwiched between the red recombinase sites. This terminator is sandwiched between the blue recombinase sites. So in the base state, both of these terminators block the activity of the transcriptional promoter, and therefore GFP is not expressed. Right, so look at the data here. In the base state, very few cells actually express GFP because these two terminators are active. And so therefore, this is the logic function we have. Basically, both inputs are 0, there's no input to the cells, and the output is 0 because GFP is off. Now, if we had one input, for example, a small molecule here, AHL, that turns on expression of the red recombinase, this produces the red recombinase shown here in red, and that flips the red triangles upside down. Right? So it leads to a circuit that looks like this. And when this terminator is upside down, it's no longer active, so it cannot block transcription from this promoter. But this terminator in the blue brackets is still active. So because it's still active, it blocks transcription, and therefore GFP is not expressed. And that leads to the logic table, truth table shown here. Now we have a second input. So suppose you add ATC and hydrotetracycline to the media. This induces expression of the blue recombinase, which flips the blue bracketed terminator. And that leads to a circuit that looks like this, where the second terminator is now upside down and no longer active, but the first terminator still remains active, and therefore we have low GFP expression as shown on here. But finally, if you had both inputs, so if you had both AHL and ATC, you induce the expression of both recombinases that will flip both of the terminators upside down, and when they're upside down, they're no longer active, and therefore the promoter can then express GFP, and therefore most of the cells actually turn into GFP positive cells in the presence of both of the inputs. And this corresponds to the truth table bottom row shown here, where you have both inputs as 1s, and therefore the output is a 1, because now you've expressed GFP. So this basically performs a logical AND gate, a circuit that only turns on when you have both inputs that are on. So with this framework, you can actually build all possible two-input Boolean logic gates. I won't go through all of these in detail here, but you can find the information in the paper citation below. But this provides a very facile way for you to program biological circuits that carry out a specific genetic logic function. The other thing this allows you to do is to encode the output of those logic functions into DNA-encoded memory. So for example, this is another example of an AND gate, and I'll leave it up to you to kind of look at why this implements an AND gate. But what we do here is we flip the circuit from off to on between day 0 and day 1. So as you can see here, GFP goes from low to high. And then we take away the inputs after day 1 and repeatedly passes the cells over 9 days, over 90 generations. And as you can see, the cells maintain GFP expression, which indicates that they maintain memory of this AND gate being switched on. And the reason for this should be obvious. Once you flip the GFP as well as the promoter, this new orientation is passed down from the mother cells to the daughter cells in a stable fashion. You can also use other ways of reading out the system. So here we've built nucleic acid probes for PCR that can detect any one of the 4 states in this other logic gate that we've built. And you can see from the graph here, the gels here, you can actually get specific bands that form in specific states based on the primer sets you use. And so the reason why we're excited about this and one of the potential applications is that you might be able to engineer cells, for example, probiotic bacteria, that contain these genetic switches. And in the future, maybe you could have patients or people that can take these probiotics, and as the probiotic transits through your gut, goes into your small intestine and large intestine, these probiotics could be engineered to sense things like inflammation or bleeding or other markers of disease, switch on a memory circuit, and when they come out the other end, for example, in your feces, they could actually generate a color or other outputs that could tell you what actually happened inside of your body. And so these could serve as new non-invasive diagnostics or non-invasive therapeutics that could sense and respond to environments inside of your body. And memory as well as logic is a very integral part of that equation. So I've told you a little bit about digital logic circuits and how you can integrate Boolean logic computing as well as DNA-encoded memory with using recombinases. In the remaining time, I'll tell you now about analog circuits that can compute information as well as analog memory devices. So analog computing is a different concept than digital. So digital, again, discretizes signals into zeros and ones, but analog computing uses the continuous concentration of a molecule to compute information. This is very similar to other forms of computing that have used analog before. For example, mechanical computers, as well as the slide rule, basically computes mathematical functions based on mechanical laws. In electronics, we also can build analog circuits. Here's an example of an analog adder, where the output voltage is a sum of the input voltages scaled by different resistor concentrations that are used in the circuit. So here, we can build an adder where the input-output relationship between the output voltage and the input voltages depends on the different resistors that are used. And this gives you a quantitative and analog way of computing information. And we would argue in most biological systems are actually analog, not digital. So many biological systems have different responses, have different concentrations of the input, which is an analog type behavior. Many biological systems, for example, sensory systems, also obey Weber's law of functions. So for example, Weber's law is this idea that the percent change in signal divided by the background remains constant. And this type of behavior is really an analog function. So for example, your ability to perceive a 1% change in light stays invariant depending on whether you're in a dark or light condition. Again, that's an analog type behavior rather than a digital behavior. So how do we build analog gene circuits? One of the ways we can do this is actually with transcriptional circuits. And so here our goal was to take an input like arabinose and log transform the arabinose input into an M-cherry concentration. So here we want to implement the mathematical function of a logarithm. So the way we did this was to actually look at a transcription factor known as ARAS-C, which activates transcription at a promoter called PBAD in the presence of a small molecule known as arabinose. So this is the original base circuit we built, and the output is M-cherry. And so when we do this, we have the circuit input-output transfer function as shown here in the red dashed lines, where the more arabinose you add, you start off actually in a low state, and then the more arabinose you add, you quickly get a switching from low to high. So this is a pretty digital circuit because the switching from low to high happens over a very narrow range, over about an order of magnitude of the input. But one of the things we noticed was that over this short switching range, this line here of the fit was actually linear. And if you notice on this graph, this is a linear y-axis versus a log x-axis, which means that a line here on the graph really corresponds to a logarithm calculation. So over this intermediate range from switching from low to high, this circuit is actually calculating a logarithm. In other words, the M-cherry output is a log proportional to the arabinose input. However, because this happens over a very narrow range, it's not actually a very useful analog circuit. And so what we did was to devise another genetic circuit that has a positive feedback loop so that arabinose activates the ARA-C transcription factor, which then self-activates its own transcription. And then the ARA-C transcription factor is furthermore on a low copy plasmid and targeting many copies of the PBAD promoter on a high copy plasmid to activate output M-cherry. And what this does, it allows you to delay the saturation of this transfer function, both with respect to the arabinose ARA-C interaction, as well as the activated ARA-C interaction with the promoter. And this allows you to build a circuit that looks like this, shown in black, where you have a pretty linear relationship between the output and the input over several orders of magnitude. And again, because this is plotted on a linear versus a log scale, a line on this graph indicates a logarithm that's being calculated over several orders of magnitude. So here with a very simple circuit motif, we've implemented a logarithm that can work across several orders of magnitude. We can also demonstrate a negative logarithm, so one that has a negative slope. And the way we do that is to take basically the same motif I showed you before, which has a positive feedback loop. Here using a different transcription factor, LuxR, where LuxR self-activates itself in the presence of AHL and targets a high copy plasmid promoter for activation of GFP. We can take the output of this circuit and use it to drive a transcriptional repressor like LAC-I, and use LAC-I to repress M-cherry, which is another output of the circuit. And when you do that, you get a diagram that looks like this, where the more AHL you add to the circuit as the input, the less M-cherry fluorescence you get as the output. And this, again, looks like a very straight line on this linear log graph, indicating that we can perform a negative logarithm, basically transforming the AHL concentration by a negative log into the M-cherry fluorescence output. All right, so what can we do? We can actually compose these circuits together. So here we've taken a positive logarithm circuit that's driven by arabinose. We take the negative logarithm circuit that's driven by AHL. And they both express M-cherries such that the total M-cherry in the cell is just the sum of the two M-cherries produced by these circuits. This basically performs the mathematical function shown above. And you can see that this is true when we look at it on a 3D graph, that the more arabinose you add, the more fluorescence you get. The more AHL you add to the system, the less fluorescence you get. And this basically performs like a plane on this graph here, which indicates that this is performing basically a logarithmic transformation. Another thing we can do with this for the subtractor circuit, remember log A minus log B is the same as log A over B. So if you tune the ratios or the slopes of the logarithmic circuits I showed you before to be approximately equal, you can actually convert the same circuit topology I showed you before into something that calculates a ratio. So this circuit here calculates the ratio, the arabinose divided by AHL ratio, and takes the logarithm of that to produce M-cherry. So as you can see here in the graph, the output fluorescence is log proportional to the ratio of two inputs, arabinose divided by AHL, over several orders of magnitude. So every data point you see here that is on the same, that has the same X value is basically the same arabinose divided by AHL ratio, but different absolute amounts of arabinose or absolute amounts of AHL. And this tells you that you can make circuits that are responsive to the ratio of two inputs rather than the absolute amounts of two inputs. And where this could be really useful is for biosensing. So imagine you're trying to sense the concentration of a molecule relative to a background. You want to make a decision based on that. You want to actually measure the relative ratio of the molecule versus the background rather than just the absolute amount of that molecule. And this is again where analog circuits can be quite useful for practical applications. So I've given you several examples of where you can do analog computing using living circuits. Finally, I'll end by telling you about analog memory. And so what do I mean by analog memory? I showed you an example earlier of digital memory where we had cells that could remember absence or presence. Did I ever see this input, for example? So that's a digital sense of information. It's 0 and 1. Analog memory refers to the idea, can I remember how long or how much of an input I've seen? Something quantitative about that signal. So to do this, we developed a new genome editing platform to encode memory into the genome of living cells. And so the way we do this is we conceptualize the the genome inside of living cells as a tape, kind of like a tape recorder, because every single base, A, T, G, and C, that's in the DNA encodes information. And so if we can write specific base pairs into the DNA of living cells, that should allow us to get very dense encoding of information. So to do this, we divide as a system in bacteria, where you have a plasmid that is induced by any arbitrary promoter, which can be hooked up to any sensor that you're interested in. This expresses a cassette that basically produces single-stranded DNAs inside of the cell. These single-stranded DNAs carry specific mutations that are targeted by the single-stranded DNA based on DNA homology to the genome, where you can then write these mutations into the genome as a way of encoding information into the genome of these living cells. And if we can induce then the expression of these single-stranded DNAs and recombine them into targeted locations, this will allow us to encode information into the DNA of living cells as a way of encoding memory. So now how do we expand this to the idea of analog memory? Well, if we make the DNA writing system one that has intermediate efficiency at writing DNA, so not too good, but not too bad, then we'll get this following scenario. So let's say we start off with a given cell that has a genome shown in red and the single-strand DNA expression cassette shown in blue, and we induce the cells over a period of time. Well, the longer you induce these cells for, the more cells will pick up a mutation in that population, such that by the end of the experiment, by looking at the proportion of cells that have that specific mutation, you can determine how long or how much of an input that cellular population has seen. So we have basically encoded the magnitude or the duration of the input into recombination frequency in a population of living cells by writing to the DNA contained within that cellular population, which can then be read out through a variety of means, as I'll show you in the next few slides. So to do this, we first wanted to develop a way to express arbitrary single-strand DNAs inside of the cell. We actually found a class of genes known as retrons. These were described about 30 years ago. This is a very simple system composed of reverse transcriptase, shown here, RT, and the MSD and MSR sequences, as shown here. And so what happens is we can put this under the control of a promoter that's inducible by IPTG, which is an artificial inducer molecule. When you add IPTG to the cells, you form the MSD and MSR sequence. This is recognized by the reverse transcriptase, which converts the MSD sequence back into single-strand DNA through a process of reverse transcription. And at the end of the day, you get this hybrid single-stranded DNA RNA molecule. So if you look on the gel here, you can see that in the presence of IPTG, we get the formation of a very nice band here that indicates we can make single-stranded DNA. This effect goes away if you don't add IPTG to induce the cells. It also goes away if you deactivate the reverse transcriptase. So you deactivate the reverse transcriptase process here, demonstrating that this is indeed a retron-dependent single-strand DNA formation. So now that we can make single-strand DNAs, how do we then add new sequences into the retron cassette so we can target anywhere we want on the genome? One way of doing that is to scan along the MSD sequence and look at where you can tolerate artificial sequences. So here we inserted an artificial sequence encoded in red into various locations in the MSD sequence. And we found that in the hairpin region, you could actually tolerate the insertion of these sequences. So as seen here, where we add IPTG and we actually insert this artificial sequence, you actually get a larger single-stranded DNA band that's formed, indicating that we've successfully encoded a new and larger piece of single-stranded DNA into the retron cassette. So now how do we use this for memory? So we can put the retron cassette under the control of a synthetic input like IPTG, and then we co-express the single-strand DNA with a protein known as the beta protein from lambda phage under the control of a synthetic promoter that's inducible by ATC. And the reason why we do this is that it's thought that the beta protein can help incorporate the single-stranded DNA into the replication fork of bacteria, and by doing so, mutate specific targeted loci depending on the homology of the single-strand DNA to the target cells. So to test this hypothesis, we actually built E. coli that actually had a kanamycin resistance gene in the genome that had 2 stop codons, thus rendering these bacteria kanamycin sensitive. And then we encoded an artificial single-strand DNA that would revert these 2 stop codons into the wild-type sequence and thus make these cells kanamycin resistant. So now by plating these cells on either kanamycin plates versus no selection plates, and by counting the percent of cells that are kanamycin resistant, we can quantify how many cells have picked up a specific mutation. And so what we show here is that in the presence of ATC and IPTG, so when you induce the retron cassette as well as the beta gene, you can get a significant increase in the recombination frequency of this population. This effect goes away if you remove any one of the two inputs, or if you deactivate the reverse transcriptase as shown here in black. This indicates that cells can record the presence of external inputs in the form of IPTG and ATC in this case into DNA encoded memory. So how do we use this now to encode magnitude or duration information? We can put the entire cassette under the control of a synthetic promoter that's inducible by IPTG. And as you can see from this graph here, the more input you add to the cells, the more cells pick up the mutations. So again, by looking at the end of the experiment and looking at the percent of cells that carry a specific mutation, we can back out how much of an input those cells have seen. And this tells us more than just absence or presence, but how much of an input you've seen. You can also encode duration information. So here we've hooked up our Retron cassette, which we also call scribe. We hook it under the control of an optogenetic circuit that is inducible by light, and then we expose these cells for 4 days either to dark or light conditions. And we find that in the dark state, very few cells pick up the desired mutation, but in the light state, so when you expose these cells to light, you can basically get a steadily increasing percentage of cells in the population that carry that specific mutation, thus telling you that basically by, again, looking at the recombination frequency in this population, you can back out how long of an input these cells have seen. So another way of looking at analog memory is to look at different patterns of induction. So here we have cells that are exposed, for example, to no input for 12 days, as shown here in black. And basically these cells don't pick up many recombinants. We also have cells shown in situation number 2 here, where we expose the cells over 12 days, shown in red. And basically these cells accumulate mutations across the 12-day experiment, as you'd expect. We also have a third case. So for example, example number 3, we expose the cells for 6 days and then we stop for 6 days. And that corresponds to the data shown in green, where you basically get a steady increase in the recombination frequency followed by plateau, as you'd expect, because we stop induction after 6 days. And then finally, in case 4, we induce cells for 3 days, we stop for 3 days, we induce for 3 days, and then we stop for 3 days. And as you can see here from the blue curve, this corresponds to a stepwise increase in the recombination frequency in the population that corresponds to the induction of these cells. At the end of the experiment, though, one of the interesting things to note is that the recombination frequency of case 3 and case 4 are approximately the same, which makes sense because they were exposed to about 6 days of input, regardless of different temporal patterns of that input. And the recombination frequency is about half of the case 2 cells, which were exposed to about 12 days of the input, as you'd expect. So now we've shown that this is a reliable way of encoding temporal information into recombination frequency in the form of DNA. So in summary, I've shown you many examples of how you can build genetic circuits to endow new computational functions on living cells. So what are applications that people are pursuing with this? The field is very interested in a wide variety of therapeutic applications, for example, developing new strategies for targeting antibiotic-resistant bacteria, as well as manipulating the human microbiome. There are other efforts to try to engineer new cancer therapies by building very responsive cell and gene therapies that can detect cancer and kill cancer. There are other folks who are interested in probing more complex human diseases using genetic circuits. Beyond the therapeutic aspect, folks are also interested in making new molecules, for example, biofuels or other commodity chemicals, where the ability to engineer genetic systems and control dynamically the output of these circuits is a useful way of optimizing production of molecules. And folks are also interested in building genetic circuits that can control the formation and the patterning of new types of materials, including things like amyloid fibers that are secreted by bacterial systems. So here's one example of a therapeutic approach that folks are interested in, and I kind of alluded to this earlier. The idea is, could you engineer a genetic circuit that senses some sort of input and triggers a response? And could you then upload that circuit into the body through a therapeutic vector? So for example, here this could be a probiotic bacteria that is introduced into a patient, and when this bacteria goes through the gut of the patient, it can sense where there's dysfunction or disease, and maybe even respond by rewiring the malfunctioning networks, by either producing molecules or tuning existing genetic networks to reset those networks into the healthy state. So I've given you an introduction into different genetic circuits that you can use to endow new functions into living cells, and giving you a brief overview of the applications that you can address. I want to finish by just summarizing my thoughts on some of the open challenges of the field to encourage folks who are watching this video to get excited about synthetic biology and the opportunities that it presented. I think if you compare the state of the field and where it's at, synthetic biology is really at a very nascent state. You know, you're comparing it to something like the semiconductor industry, we're really just in the early inflection point of what we're going to be able to achieve by being able to program biological systems if we can tackle many of these open challenges. And so one of the key challenges is trying to be able to design genetic circuits that operate in the confines or in the context of very complex biological systems that we don't fully understand. So this is, these are diagrams that kind of symbolize the complexity of natural biological systems. People oftentimes refer to these as the hairball of biological networks, where you have, regardless if you're looking at transcriptional networks or protein-protein interaction networks, in natural biological systems, everything seems to be interwired, and it's very hard to discern what are the design rules that are needed for biological systems to function robustly. So then this raises another question of how do you design biological systems in the context of these networks where we want our circuits to be robust and not be necessarily negatively influenced by these networks, but we also want our circuits to be able to interface with natural networks to perturb natural biological systems. So I think this is an open challenge that we're going to need a lot of effort to try to understand how do we build stable interfaces with natural biological systems, but how do we also try to insulate ourselves from the variations that you can find in natural biological systems when we don't want them. Another major challenge is how do we deal with defining programming languages and robust paradigms for building genetic circuits. So I mentioned early on how there's been exponential improvements in our raw ability to read and write DNA. This hasn't really translated into much more complex circuits. So this is a study that was done many years ago, looking at the complexity of genetic circuits by counting the number of parts that went into those circuits. And we certainly haven't seen an exponential explosion in the complexity of circuits. Even though this diagram is a little bit outdated, we have not seen an exponential increase in our ability to build complex circuits that function properly, even though we have the raw capability to read and write DNA much more than we did before. Similarly, we have not seen an explosion in new genetic circuits and devices that have been described. And part of this is that we still haven't fully understood what is the right programming substrate, what are the right primitives, and how to put together the devices into more complex circuits to get more complex functions going. And we need more efforts to try to understand what are the programming rules for building biological systems from the ground up. And so to end, I wanted to pose some challenges and bottlenecks in the current synthetic biology design cycle. We often like to think of kind of going through this chain of events to design a system, but as I'll show you a little bit later, this is really challenging to do currently. So many people currently conceptualize designs. People go to the board and draw these by hand. We'd like to be able to have a more rigorous way of designing functions. Is there a language by which we can describe biological circuits? And then is there a way to then compile that high-level abstract concept into downstream circuit designs? So that's one major area that folks are working on. In the area of actual implementation of designs, one of the major challenges over the last few years has been that many of the parts we use are the same parts reused over and over again. There aren't that many super well-characterized systems. And I think as people move to new scaffolds or engineer libraries of artificial parts, we're going to be able to expand the library of well-characterized parts that we can use to build a variety of biological functions. Another major challenge has been that our modeling and simulation frameworks are largely not predictive of experimental results. And as a result, we can't use solely the computational algorithms to build new biological systems. So this remains a major challenge. What are the right modeling frameworks to model the performance of biological systems? How do we make them accurate? And how do we make them predictive? And similarly, if we use simulation, how do we make these simulations fast, robust, and predictive? On the construction level, there are many different new techniques to now enable the rapid assembly of more complex systems, but this remains a pretty laborious process. So many times people spend a long time trying to build their system and much less time conceptualizing or designing the system. And this focus on experimental construction would be great if we can lessen the dependency on kind of physical construction of DNA. This would really greatly accelerate the design, build, test cycle. And similarly, new technologies for probing and measuring biological systems are necessary to really be able to accelerate this whole engineering cycle. Finally, I alluded to this early on, but this challenge of evolution. So we can use evolution to optimize our genetic constructs So for example, you can kind of build a genetic construct that doesn't work super well, but by using evolution coupled with directed selection, you can actually improve the behavior of those circuits. But similarly, once you've designed a circuit that carries out a function, how do you make sure the bacteria does not evolve away from that function? That remains another existing challenge. So as I mentioned, the current design cycle is not like what I showed you before. Oftentimes, because the modeling and simulation frameworks are not predictive, they're used in a post hoc fashion. And we spend a lot of time really focused on construction, probing, and measuring. And so new technologies to accelerate this process, to incorporate evolution and deal with evolution will be much needed. And similarly, new frameworks for doing predictive design, modeling, and simulation will be really needed, with the ultimate goal that we can spend more time on conceptualization and design and comparatively less time on the physical construction and testing of these circuits. And that would be a great boon, I think, to the field of synthetic biology. It would enable many new capabilities that cannot be addressed currently just because it's too time-intensive or labor-intensive to implement. So in summary, I've told you about many different areas today. Synthetic biology is an emerging discipline for engineering new functions into biological systems. This will open up new application areas, including therapeutics, diagnostics, industrial applications, as well as basic science applications in terms of understanding biology. Significant advances in DNA sequencing and synthesis are already driving new approaches to engineering biological systems, and I think this will hopefully continue. I told you about synthetic gene circuits and how they're really crucial to implementing sophisticated behaviors in living cells. I've also talked about how recombinases can be used to create integrated digital logic and memory circuits by flipping memory registers located on DNA, and for doing that in a modular fashion so you can control gene expression. Furthermore, I've told you about analog circuits that can actually compute continuous information and continuous calculations like logarithms or division or addition inside of living cells. These analog circuits are very parsimonious. They don't require a lot of parts. And that's another benefit when you're trying to engineer biological systems that don't have infinite numbers of parts to go around. Furthermore, I talked about the scribe system, which enables dynamic genome engineering, and that allows you to encode analog memory into the collective DNA of living cells populations. So encoding temporal or magnitude information into the DNA of living cells. And finally, I've talked about some of the open challenges in the field, and I'd like to encourage those who are watching the video to think about new and creative ways to tackle this problem of programming biology. How can we figure out what are the design rules, and how do we make the whole design build test cycle more efficient and more robust and more predictive? And that's what we really need to make this a true engineering discipline. And so with that, I'd like to thank you for your time, and I'd be happy to take any questions. My email is listed here in case you want to reach out and contact me. And I hope you enjoyed this talk. Thank you.